Hello, biology class. This is the content video for Unit 2, Section 2. Our learning goals for today are to identify the functions of organelles, so what each organelle does, as well as to classify the organelles of plant, animal, and fungal cells. We're going to be doing that by mainly looking at the differences between plant, animal, and fungal cells. So, when we talk about organelle functions, we need to first talk about what is inside a cell. So the interior of a cell is filled with cytoplasm, which is kind of like jelly or jello or jam. And we use the term semi-fluid because it's kind of a liquid, but it's kind of not. Fluid means it can be poured and it has some viscosity to it, so it sticks together. And it fills the empty space of a cell. So that empty space does have things in it. It's just that those are molecule level, very, very tiny. And I want to remember that prokaryote versus eukaryote thing we talked about last video, which was the video from somebody else that I had you guys watch. Prokaryotes, those are the ones without nucleus, without a nucleus, so without nuclei. That's the plural, nuclei. The chemical reactions that are important to the life of the cell all just happen in the cytoplasm. They don't happen necessarily in a specific location. Sometimes they do, but in general, it just happens inside the cytoplasm. All right, the cytoskeleton is the support. Just like your skeleton is the support for your body, the cytoskeleton is support for the cell. So cyto, like cytoplasm, right? Cytoskeleton. So structure and support. Just like your bones allow you to move because the muscles pull or push on them, usually pull, the cytoskeleton acts as anchor points for the cell to move around or to move things around the cell. All right? So cellular movement, we're not just talking about moving the cell, we're talking about moving the things inside the cell. Microtubules are really, really tiny tubes, and microfilaments are really, really tiny, kind of like strings. Filaments are kind of like a string or a thread that are inside it. On this picture to the right, you can see the green lines are the areas where these microtubules and microfilaments have been rendered or modeled. Remember, this is one of those models. It's not an actual picture of a cell. So we've got the nucleus in the center. The cell membrane is the red stuff all along the outside. And this green is the microtubules and microfilaments. Something I also want to talk about here is something called a centriole, which is just a group of microtubules that do a specific function. And that is to help with cell division. So when a cell splits. We will talk about that more later in this unit. Remember that eukaryotes have a nucleus. So this nucleus is where they store their genetic material. That material is called DNA, deoxyribonucleic acid. DNA, deoxyribonucleic acid. So the nuclear membrane surrounds the nucleus, right? NuCl and UCLE, right? Same letters. They're related. The membrane goes on the outside. It looks like this, okay? You don't have to memorize this whole picture. I'm just using it to show you that it is a double layer membrane, just like the outside portion of the cell membrane. It's got these pores or channels or holes in it that allow things to pass through the nuclear membrane, but not everything gets to pass through the nuclear membrane, all right? So the big name for these are nuclear pore complexes because they are multiple things working together to function as a pore, a hole, right? The nucleus is where the DNA is gathered, and it functions to protect it and to make ribosomes. So ribosomes are made inside the nucleus. A ribosome helps make proteins. Proteins are what allow your body to do things on a very, very tiny level. All right. There are two types of ribosomes: free floating, which are just floating throughout the cytoplasm, And they can also be bound to the endoplasmic reticulum, which is called ER. The ones that are bound to an endoplasmic reticulum make the endoplasmic reticulum a rough endoplasmic reticulum. Remember, they are made in the nucleus. All right, so that does lead us to the endoplasmic reticulum, which is kind of a weird sounding word. It just is a folded system of sacs. So instead of being a double layer, this is generally a single layer, right? And this diagram here, this figure, shows you how it's connected to the nucleus. The ribosomes are on the rough endoplasmic reticulum, which is this red part, and this orangish part is the smooth endoplasmic reticulum because it doesn't have ribosomes, all right? So this works to make proteins in the rough endoplasmic reticulum and lipids, which are kind of like fat cells, in the smooth endoplasmic reticulum. So lipids is a term that can describe fat cells or fatty acids or fats. Okay, I know that's kind of complicated. Don't worry about it too much. Just for now, remember that the smooth ER makes lipids, the rough ER makes proteins. Remember, because we're talking about functions here. We want to know what they do. The Golgi apparatus, which, yes, it does sort of look like this, and it looks weird, I know. It modifies, so it changes them a little bit. It sorts them, so it makes them into like stacks or classifications. And packages, so puts them together and stores them in specific locations, right, in vesicles, of those proteins. So the proteins, right, we know that the nucleus makes ribosomes, and ribosomes make proteins. Those proteins then go to the Golgi apparatus and get put into vesicles, all right? And we know that vesicles are a special membrane sac that can merge with the cell membrane, so they actually can go to the cell membrane and merge with it, so that means to become one, right? And push its contents out of the cell, so it can transport these proteins out of the cell. So this is kind of like a shipping center, like UPS or FedEx or the post office. Similar to a vesicle is a vacuole. All right, they even sound similar. Vesicle, vacuole. Both start with this V sound, right? So this is usually only in plant cells. You can tell this is a plant cell because it's green, right? No. We're also looking at this thing called a cell wall. We'll get to that. But it's a large because we're still on cellular level. And it is a big place to store food, enzymes, which are a type of protein, water. Water is important to everybody, even plants, especially plants. And a couple other things you don't need to worry about. This food here, what we're talking about is chemicals, not hot dogs and hamburgers, right? Plants don't eat that. This is a lot of starches, too. All right, so vacuole is storage for food, enzymes, and water. A lysosome, or lysosome, is kind of a digestive system for the cell, but it doesn't digest food to make energy. It digests things to break them down and get them out of the way. So looking toward lysosome, lys means to split or cut, right? That goes back to Greek. So lys is lys, to split or cut, to separate. And this just digests, so breaks apart waste, so things that the cell doesn't need anymore, extra or old organelles. So these are organelles that are past their prime. They are wearing out. They are being replaced. So the old ones get destroyed. Now those destroyed organelles, what made them up, can then be used again and recycled inside the cell, or it might be transported outside the cell. Right? The lysosome has its own membrane because the digestive enzymes, the proteins, remember enzymes are just proteins, inside it could be harmful to everything else inside the cell, just like your stomach has a lining to protect the rest of your body from stomach acid. Mitochondria, yes, the powerhouse of the cell, Don't remember powerhouse of the cell. Remember that it makes adenosine triphosphate through something called cellular respiration. Cells don't breathe. You breathe, right? Cellular respiration is more like food digestion, but on a cellular scale, so very, very tiny. And this is converting the chemicals from food, so that hot dog, hamburger, tofu, whatever you're eating, and oxygen that you breathe into energy that the cell can use. Remember, cells use energy, you use energy, right? But you don't just eat energy, you eat food. Chemical energy, right? And that is called adenosine triphosphate, ATP. This might as well be a vocab word. I may add it to the vocab list because it's important. It's going to keep showing up this unit, right? The mitochondria has a couple of membranes, right? It has this interior membrane. You can see it as these thin orange lines here. They're kind of a lighter colored orange, and they're all squiggly, right? They're mushed together and folded. That gives it a much bigger surface area, an outside covering, right? But it's on the inside. This large surface area allows it to have more chemical reactions. More chemical reactions means more ATP. Okay? So higher surface area, more reactions. More reactions, more ATP. More ATP, more energy, and more stuff that the cell can do. That interior membrane is covered by another exterior membrane. So the mitochondria has two membranes. This is one of the reasons that we think that mitochondria actually came from outside the cell and was enveloped by the cell a long, long time ago. Like billions or even trillions of years ago. All right, we are talking old school stuff. Switching from animal cells to plant cells have chloroplast. Chloroplasts are these tiny green things in the cells over here, and they absorb light energy, remember solar light, sun, and convert it to chemical energy. Chemical energy is just the energy that they need, right? They're not using electricity, they use chemical energy. And so chloroplasts store chlorophyll pigment, which is green. The chlorophyll is what actually absorbs this light energy. And we'll talk more about that later. Plant cells also have a cell wall, which is a kind of exterior wall or mesh of cellulose. Sometimes pectin is also inside it, that covers the plasma membrane. So remember, rigid mesh of cellulose covers plasma membrane. So it's on the outside of the plasma membrane. It gives protection and structure. Plants don't have bones, right? They rely on this cell wall to give them structure. Two last organelles. I know we've had a lot. This video is probably going to hit 15 minutes. Cilia and flagella. So cilia are short protrusions on the exterior of cells. They kind of look like hairs. And flagella, which are long protrusions on the exterior of a cell, they kind of look like whips or a tail. And they do this. Okay? So both of them allow the cell to move. The cilia can also move things across the cell. In your small intestine and large intestine, the cilia on the outside of the cells there allow the digested food to continue moving through the intestines. All right? So their function is to move the cell or other things across the outside of the cell. All right, wrapping up, differences. We have our animal cells, our plant cells, and our fungal cells. Animal cells do not have a cell wall. They do have mitochondria. They do not have chloroplasts. And then this last row that's in italics have to do with chromosomes. I know we haven't talked about chromosomes yet, but we will. So usually diploid, so di means two. So they have two pairs of chromosomes. Plant cells have a cell wall that contains cellulose. They usually don't have mitochondria. They do have chloroplasts, and again, they are usually diploid, two pairs of chromosomes. Fungal cells, so that's fungi, mushrooms, lichen, that kind of stuff. They have cell walls, but they have chitin and proteins in their cell walls instead of cellulose. So that's the difference in their cell walls. They do have mitochondria instead of chloroplasts, and they are usually haploid. So if diploid meant 2, haploid means 1. So one pair of chromosomes instead of 2 pairs of chromosomes. Here is a quick 3D, sort of 3D. I know it's a single flat image, but it's got some perspective to it, so we're going to call it a 3D picture of a animal cell, right? We know this is an animal cell because it has no cell wall, and it has these things. These are mitochondria. A single mitochondria is called a mitochondrion. I know that doesn't follow the normal English rules of single to plural. That's just the only one that you have to worry about, though. Okay, so a single mitochondria is called a mitochondrion. You can see the nucleus. Inside the nucleus, in this one, has a nucleolus, which I didn't talk about earlier. That's because it's not something I need you to focus on remembering. I'm more concerned with you remembering the nucleus. The nucleolus is the specific gathering of DNA inside it. So it's a denser structure inside the nucleus where the DNA mostly resides. We've got all these little dots, which are ribosomes. Some of them are free-floating out in the cytoplasm. Some are attached to the endoplasmic reticulum. On the outside of the rough endoplasmic reticulum, we have the smooth endoplasmic reticulum. We have some centrioles floating out in the cytoplasm, as well as the Golgi body and lysosomes. All right? So it might be a good idea for you to actually take this image. You can screenshot it off of this slide. You can copy this slide off of my slides and study it. Maybe scratch out these labels and try to do the labels on your own. I can provide a blank one of this if you would like. Send me an email about that. All right, so our 2 learning goals, right, to identify the functions of organelles and to classify the organelles of plant, animal, and fungal cells. We went over each type of organelle and its function, and we talked about the differences in the cells so that we would see where those organelles were and were not. Big difference there were mitochondria, chloroplasts, and the cell wall. All right? If you have any questions, as always, send me an email, and you can drop into my office hours. Those are Monday through Thursday from 2 to 3, and on Friday from noon to 3. If you swing by in the last 10 minutes, I may be packing up for the day to head home because I got to pick my daughter up on the way home. Have a good one.